Dear students, let us take another topic, that is genetic engineering process and what are the steps which are involved in rDNA technology. Let us take the definition of genetic engineering process. Genetic engineering indicates direct manipulation of genome of a cell. We can call it as genetic engineering. The work of this genetic engineering was done by Palberg, and the honor of his work, right now we are calling him as a father of genetic engineering. What are the steps which are involved in genetic engineering? So the first step is isolation of DNA fragments from the different types of cells. How can we isolate different types of enzymes which plays an important role for the isolation of genes? You can take plant cells. With the help of cellulase enzyme, we can able to isolate gene or DNA from the plant cells. And you can take chitinase. So chitinase is from the fungi cells, and you can take lysozyme. So lysozymes are the enzymes which we use for the animal cells. These three different types of enzymes which are highly essential for the isolation of DNAs or DNA fragments. So cellulase for the plant cells and chitinase for the fungi and lysozymes for the animal. So based on this enzymatic action, the walls can be get degraded and we can able to isolate the genome. After isolation of genome with the help of restriction endonuclease enzyme activity, we can able to break down it and the DNAs are ready to be transferred. So such DNAs can be cloned very easily. Such DNAs are also called as DNA inserts. And if you take regeneration of number of DNA strands by introducing the strands into the other DNA molecules. If a DNA is having ability to carry other DNA molecules for replication, such kind of DNA molecules we can call it as vectors. Vector is nothing but it's the simplest word is a DNA molecule which is having ability to carry the number of DNA strands for the manipulation or multiplication in host to cell. Self replication takes place within a host to cell. So that kind of DNA is called as vectors. You can use plasmids, cosmids, phasmids, bacteriophages. This all can act as a vectors. Plasmids are the very well known vectors for the cloning process. And if you take these vectors, you can identify two types of vectors. Vectors can be divided into the two types. One is cloning vectors, another one is expressive vectors. If you take cloning vectors, number of copies can be produced, and if you take expressive vector, recombinant proteins can be formation takes place. Plasmid PBR322, you can take it as a cloning vector, and expressive vector PUC is one of the best example. And after selection of this vector, what we'll do and what is the procedure, and let us follow the diagram. With the help of diagram, we can able to understand very clearly rDNA technology. So first we need to isolate the plasmid from the bacterial cell, that is Escherichia coli. So our target genome is here. Directly you can take DNA from the cell with the help of lysozyme, or you can take chitinase enzyme, or you can take cellulase. So different types of enzymes will degrade this wall, and directly we can able to isolate the DNA. So this isolated DNA, and after that with the help of restriction endonuclease enzyme, we can able to do fragmentation. So once you do the fragmentation process, this here there are number of insertional sites. You can cut the plasmid at the insertional site, and you can tag this this DNA, that is desirable DNA, into the plasmid DNA. Then it will become as a single DNA molecule. This single DNA molecule, now if you observe here very clearly, a DNA contains our target DNA. So a DNA is formed by the joining of two different types of DNAs from two different individuals. We can call it as plasmid DNA, and this plasmid DNA also called as vector DNA. And this vector DNA, recombinant DNA, vector DNA or recombinant DNA, directly transfer into the host to cell, whether it is homogeneous host or heterogeneous host. You can take the cell from where we have extracted, or else you can transfer into the new kind of bacterium. After this, what will happen? Number of cells, this DNA involved in replication, self-replication process, because plasmid abilities, selectable markers, and ORI site and cloning sites. With the help of that ORI site and cloning sites, it can be involved in manipulation and multiplication. At every 20 minutes, it will divide and it will form more number of our foreign DNAs. So for the identification of our foreign DNAs, we can use selectable markers method, and after using selectable marker method, we can able to separate desirable DNAs and we can transfer into the host cell. We can get a desirable DNA fragment. Else you can transfer directly to the bioreactor and you can get recombinant proteins. So the major steps which are involved in this process, first step is isolation. So isolation of genome. Second step is second step is isolation of genome from the bacteria as well as host cells. Second cell is construction of rDNA. Construction of rDNA. So we should construct rDNA. After construction of rDNA, insertion of this rDNAs, recombinant DNAs into the host cell. So construction of rDNA, insertion of this rDNA into the host cell. After insertion of this rDNAs into the host cell, manipulation or multiplication takes place. Manipulation or multiplication takes place. After manipulation or multiplication, directly we can able to extract. So extraction of desirable DNA fragment based on selectable markers. So based on the selectable marker, we can able to isolate desirable one, and you can transfer into the host cell directly. So after transferring into the host cell, this host cell directly placed on culture medium, and it gives rise to the new individuals. Or else you can use bioreactor. So when you transfer this desirable DNA fragment and when you place in bioreactor, so it leads to the formation of number of desirable DNA fragment, that is called as recombinant proteins. So bioreactor is a stainless steel vessel which is helpful to formation of number of product. So recombinant product can be obtained. This is about the bioreactor. Hope you understand. Thank you.